Autophagy plays a crucial and multifaceted role in limiting the spread and impact of senescent cells, those dysfunctional cells that stop dividing but don't die. Senescent cells are often described as zombie cells because they linger in tissues, secreting harmful inflammatory factors, enzymes, and growth signals collectively known as the senescence-associated secretory phenotype, SASP. Over time, this toxic secretome degrades the surrounding environment, disrupts neighboring healthy cells, and accelerates tissue aging. The accumulation of senescent cells is strongly associated with chronic inflammation, fibrosis, loss of regenerative capacity, and a variety of age-related diseases. Autophagy, as the body's cellular renewal mechanism, serves as a critical line of defense against this downward spiral. One of the primary ways autophagy limits cellular senescence is through its preservation of intracellular homeostasis. By clearing damaged mitochondria, misfolded proteins, and oxidized lipids, autophagy keeps cells metabolically balanced and free from the kinds of damage that trigger the senescent switch. When autophagy is functional, cells can endure stress and repair themselves, maintaining function and avoiding the point of no return into senescence. But when autophagy is impaired, damaged organelles and toxic metabolites build up, pushing the cell toward dysfunction and a permanent arrest in the cell cycle. Notably, autophagy and senescence are not strictly opposing processes. In some contexts, they are tightly interwoven. In early stages of cellular stress, autophagy acts protectively, promoting survival and recovery. But when stress becomes overwhelming or prolonged, senescence takes over. Interestingly, autophagy can persist even in senescent cells, not to save them, but to support their secretion of SASP factors. However, research has shown that certain types of autophagy, particularly mitophagy, are reduced in fully senescent cells, contributing to the increase in ROS and inflammatory signaling that characterizes their behavior. The more promising insight is that boosting autophagy may not only prevent cells from becoming senescent, but also help the immune system recognize and clear existing senescent cells. Emerging studies suggest that autophagy supports immune surveillance by enhancing antigen presentation and reducing immune exhaustion. Natural killer, NK, cells and macrophages rely on autophagic flux to maintain their cytotoxic activity and phagocytic function. When these immune cells are robust, they are better equipped to identify and eliminate senescent cells, acting as a cleanup crew that removes the lingering threats before they disrupt tissue structure. There's also compelling evidence that autophagy influences epigenetic stability, another factor in whether a cell becomes senescent. Senescent cells often show widespread epigenetic dysregulation, including histone loss and chromatin remodeling. By preserving the quality and turnover of nuclear components, autophagy contributes to a more youthful epigenetic landscape, protecting against the genomic instability that can push a cell toward the senescent phenotype. From a therapeutic perspective, interventions that stimulate autophagy, such as intermittent fasting, calorie restriction, spermidine supplementation, or pharmacological agents like metformin, have been shown to reduce the burden of senescent cells in aged tissues. This is not necessarily because autophagy directly kills these cells, but because it improves the overall tissue environment, prevents the seeding of new senescent cells, and helps immune mechanisms work more efficiently. Over time, this may significantly delay the functional decline associated with aging organs. In summary, autophagy acts as a guardian against the cellular decay that leads to senescence. By maintaining internal order, reducing oxidative burden, supporting immune vigilance, and sustaining epigenetic fidelity, it not only delays the formation of senescent cells, but may also help contain their spread and toxic influence. In doing so, autophagy protects tissue integrity, extends regenerative capacity, and preserves the quiet coordination of cellular life that defines youthfulness. As research deepens, it is increasingly clear that keeping autophagy active is one of the most effective strategies for limiting the harmful reach of cellular senescence and for promoting a longer, healthier life.